So we now move to our keynote presentation. So we're very, very pleased to have with us Jocelyn Millar. Jocelyn Millar is internationally known for his research on insect chemical ecology and the development of applications for insect semiochemicals and related compounds. His research in insect chemical ecology is vertically integrated to encompass basic behavioral studies demonstrating that chemical communication does occur, the isolation, identification, and synthesis of chemical signals, and the verification of the biological activity of various components or blends. These studies have extended the development of practical applications that use insect pheromones and related chemicals for detection, sampling, and management of insects. Much of his work reflects the multidisciplinary research areas that are involved in the various components. He was born in England in 1954 and immigrated to Canada in 1957. He was out west attending the University of British Columbia for 2 years, studying engineering, then worked and traveled in Europe, Africa, and Asia for 2 years. So lots of travel like our past winner. After returning to Canada, he enrolled at Simon Fraser University, graduating with a bachelor's in chemistry in 1978. He obtained his PhD in 1983 in chemistry at Simon Fraser, identifying and synthesizing grain beetle aggregation pheromones. After graduation, he worked on host plant-based attractants for elm bark beetles for a year. He joined the National Research Council of Canada laboratory in Saskatoon, Saskatchewan for 2 years, studying pheromones of geometrid and artid moths. He then ran a large toxicology lab in Vancouver for 2 years and then left us to went to the University of California, Riverside in 1988. Millar has published more than 275 scientific papers, 24 book chapters and review articles, and edited 4 books. He has presented and co-authored more than 200 invited, 300 submitted presentations at state, national, and international conferences. His list of awards will take us the rest of the afternoon, so I am highlighting a few. He received multiple awards from the European Space Agency since 2001. He was elected a Fellow of the American Association for Advancement of Science in 2003, and he was selected as the 2014 Silver Medal winner by the International Society of Chemical Ecology in recognition for his career achievement in chemical ecology. Please join me in a warm welcome to Dr. Millar. His presentation title is Travels with My Ant, Deciphering the Chemical Languages of Insects.
It's a great pleasure to be here. It's a great pleasure to be back in Canada. I'm still very much a Canadian citizen, and so I enjoy coming back whenever I can. What I'd like to talk to you about today is basically the travels that I have made through the chemical world and the chemical ecology world. And I hope you forgive me I'm totally addicted to bad puns. And so Travels with My Ant, possibly the younger people in the crowd might not know that this is a famous book by a guy called Graham Greene. And if you look it up on Wikipedia, it's described as Aunt Augusta pulls Henry away from his quiet suburban existence into a world of adventure, crime, and the highly unconventional details of her past. So today I'm hoping I can tell you something about some unconventional signaling systems that are used by various insect species. So I'd also like to point out, because this is showcasing interdisciplinary work, that a lot of my work has been done with collaborators, particularly biological or entomological collaborators. There's no possible way we could have done all it, or I could have done all this, my group could have done all this by ourselves. So there's been hundreds of people now that I've been fortunate enough to work with over the years, on a wide variety of insects. These are just a few of the various different types of insects that we worked on, and we'll talk about some of their stories as we go along. So, unlike us, insects communicate primarily by using scents and tastes. Some of them do use acoustic communication as well and visual signaling, but primarily they are smelling things and they are tasting things. So what I want to go through today is some instances of what are they saying, how are they saying it, and why is it important to understand this. And just to quickly show you, this is a male moth, and you can see here his antennae, his sensor apparatus. These are probably some of the most sensitive biological sensors that we know of. And we can take advantage of this by actually doing what are called electroantennograms, and I'll show you some of those as we go along. So where it all started for me was with these small insects. They're called grain beetles. You can get an idea of the size there from this one sitting on a kernel of wheat. And some of you may have had these as uninvited guests in your pantries because they get into a wide variety of stored products and milled flour and things like that. This was my graduate work with Cam Oschlager at Simon Fraser University. And as a chemist, I was entirely unfamiliar with the basics of doing bioassays and in fact what bioassays meant, and I hadn't really thought very much about this at all, but I was doing all of my own bioassays. And to give you an idea here, this is what's called an arena bioassay, just a sheet of paper. Each of these little black dots is one of the beetles. I had originally released them in the center here. And this is where the airstream is coming across, and on this piece of filter paper I had put one of the test stimuli that I was testing to see the responses of these beetles. And as you can see here, these beetles are essentially randomly scattered over this page, or the arena here, indicating that really there is nothing there that those insects are interested in. Conversely, this is a close-up shot, and the stimulus, you can just barely see the end of the tube here. And having released the insect somewhere around here, you can see that all of them are essentially lined up and heading towards or at least they're all lined up. Some of them are actually heading away, but they're still in this plane here. They're all lined up because they can detect that stimulus, and it's powerful enough that it's directing them all to go towards that particular stimulus, the pheromones the aggregation pheromones that they produce. So for the two different species that I worked with, these are the molecules that we eventually worked out were components of this chemical message. And you can see here they're related species, they're in the same genus, and you can see that they share one compound here, but basically the structures are really quite similar. Because they are closely related to each other, they're producing similar compounds, but they've diverged enough that each species has its own slightly different chemical message that it's using for aggregation. So what are they saying? The males are producing these aggregation pheromones that attract both sexes, and it's important because the aggregations of beetles actually modify the environmental conditions in which they live. So having a large aggregation like this encourages the growth of fungus, and they feed on the fungus that is generated. It generates heat, which is crucially important because if they're infesting, for example, in grain silos out in the prairies here, as you can imagine, unless it's warm, they're going to get killed off by the cold. And of course, it presents opportunities for mating as well. And so for us, we can exploit this attraction then to be able to monitor these insects, to be able to sample them, and in some cases possibly control them as well by being able to pull them in to either some sort of a trap or a toxic bait or something like that. So the overall take-home message for me as a chemist from doing these bioassays was to really get an appreciation for the roles of the biology and ecology as well as just the chemistry. And a message that I still try and beat into my own students these days is the importance of having a reliable and robust bioassay that can direct you as you go along on the chemical fractionation process. If you don't have that, there's no point in even starting the chemistry, because you'll just be completely at sea. It also taught me the extraordinary power of some of these chemical signals as modifiers of insect behavior, and also the importance of details. That is, you need to get these biological details right if you're going to be able to have the bioassays work and have the insects behaving the way that they would naturally behave. So things such as the rearing density of those insects, it affected pheromone production. The rearing density also strongly affected the response. If they were in high density where they didn't need to produce aggregation pheromones anymore, then they stopped responding to them because they were already aggregated, for example. The importance of synergism between these compounds. So each one of these compounds alone was not active. And so you had to essentially have the whole chemical message there in order for the information content to get through. And for the chemists in the crowd here, this was one of the first instances of synergism between enantiomers. That is, you have a right and a left-handed form of a molecule. And for these insects, you had to have both enantiomers present in order to get a response. To give you a quote from one of my favorite people, Yogi Berra, the baseball player, you can observe a lot just by watching, which I think is one of the key messages from that experience of mine as a graduate student, just learning to observe and learning to how much you can figure out from just really critically watching what your test organisms are doing. Now, this is moving on. This is some work from some work that I did in California. And somewhere in here, I think right up in here, there's a little piece of rubber which we have impregnated with the sex pheromone of these moths. These are all male moths, and they're all responding to the sex pheromone message that we've been able to duplicate. And you can see basically that you've got this huge aggregation of these moths. They all think that there's a female there somewhere, and they're just desperate to get to her. So before you put out that rubber piece of rubber with the chemical message on, you could not see a moth anywhere. 2 minutes later, you get a huge aggregation like this. They just come out of nowhere. So what it really drives home is how powerful these chemical messages can be and how they can be completely irresistible to insects. And that kind of power is something that we can exploit and manipulate for our own purposes for insect control, for example. So in this case, what are they saying? Most female moths produce these powerful sex pheromones to attract males. Why is it important? It's really the key to be able, for them to be able to reproduce. That is, as you can imagine, you've got two moths that are separated by 50 meters. They'll never bump into each other randomly, so they're using these signals to bring each other together. And we can clearly exploit this, as I've just shown you, in terms of the power of that attraction. So over the years, my group has worked on a whole bunch of different moths, from little tiny ones. This one's a couple of millimeters long to great big ones. This one's about the size of your hand or so. And these are some of the chemical messages they're producing. These are the female sex pheromones of these different species. And just in passing, you can see that these structures are really quite similar. They both have an aldehyde function. They've got a chain length that's about the same length. They've got a number of double bonds scattered along the chain here. And how can we exploit these then? Well, this first one that I showed you, this little tiny one, is actually an invasive species. It got into California in particular, but it's throughout the States now and a number of other countries. And you can see the kind of damage that its larvae cause by mining in the leaves. Basically, the foliage becomes all crinkled up, like photosynthetic efficiency of the plants goes way down. Another one that we've worked with is something called avocado seed moth. It's in Mexico, Central America, and South America. California has an avocado industry which is worth approaching $1 billion. And we do not yet have this insect, and we absolutely do not want to get it. You can see here the nasty mess it makes of an avocado fruit. So using these compounds then, we can have very sensitive methods of detecting these insects when they come in. So for other lepidopteran species that are endemic, what we can do is we can use these pheromones, put them in traps and so forth, so we can get an idea when the insects are there, approximately how many there are, and consequently we can make management decisions as to whether or not to use insecticides, for example, based on whether there's a need to, rather than just applying insecticides on a calendar basis, which is what growers used to do. So it really helps us to fine-tune the insect management for a number of these pest species. And this one is, it's called navel orangeworm, but surprisingly it's a pest of almonds and other nuts. Don't ask me who called it the navel orangeworm. And this spruce coneworm is a project we worked on with some people here in British Columbia, actually. One of the other things that we can do, sometimes at least, is we can do what's called mating disruption. And that is we use these insect pheromones. If we broadcast them in the atmosphere of an orchard or a vineyard, for example, wherever these insects are, we can essentially create a blanket of pheromone which prevents the male insects from being able to find the females. Now, that development of that type of strategy is in progress right now for the citrus leaf miner. It's a relatively recent invader of California, so the details are still being worked out. But for codling moth, which you have here in Canada, and it's a worldwide pest of apples and other pome fruit, mating disruption has now been used for a number of years, and on the west coast of, or the west United States, I believe that mating disruption is actually the most used method for controlling codling moth now. It's a successful strategy that is working very well, and it's entirely environmentally friendly. The amounts of pheromone that you're using are very small, and the pheromone itself is completely non-toxic. One of the other things we can do is we can use these pheromones to be able to monitor some endangered species, and this fantastic animal is called the Spanish moon moth, Graellsia isabella. We worked with a European group in this case who are interested in finding out where this thing is, what its range is, and because it's nocturnal and lives in pine forests, it's otherwise very difficult to keep track of and try and even gain estimates of what the population sizes are. But with these pheromone lures, as you can see here, the males are very strongly attracted, so you can get a really good idea as to whether they're there, and if they are there, how many there are. So we're continuing to work with a number of the other lepidopteran species. This is one we're working with Jeremy Allison and Sault Ste. Marie on Luna moth. Again, another gorgeous creature. And some other endangered sphingid moths in California, and some calosamia, which are some of the really large moths, the big beautiful ones. Just to, again in passing, to mention the similarities in the words in these languages, if you will. So for example here, these are all moth pheromones of one kind or another, but you can see that they're really quite similar. There's slight differences in the functional groups. There's differences in where the double bonds are and triple bonds in some cases, for example. But they're all using pretty much the same motifs, and as we know it right now, there are approximately 4 of these major motifs that different types of Lepidoptera use, broken down on the basis of taxonomy. As they cluster into these different groups. But we have a pretty good idea now as to the chemistry of these Lepidopteran pheromones. As Yogi Berra again said, it's deja vu all over again. In terms of once you've worked with one of these Lepidopteran pheromones, you have a really good idea as to what to look for. And these days, in some cases, just from a single female moth, the analytical techniques, the analytical chemistry techniques are good enough that we can determine what the pheromone is from a single female. Now, I'd like to switch over now to talk about stink bugs. And the stink bugs are so named because they're very well chemically defended. I'm sure you've all picked up one of these and squashed it or accidentally, for example, and you get the very pungent smell from them. But in addition to those chemical defenses, they also produce chemical messages to talk to each other. And it turned out that they also use some acoustic signals as well. So these are some of the chemical messages that they're producing, actually based on, to a considerable extent, on what are called sesquiterpenes, 15 carbon compounds, and then this odd one out here, which is an ester compound. And again, the idea that the chemistry of, or there are similarities in the chemicals that these related insects are producing. So once you get an idea of what one of them is producing, it gives you a really good idea what to look for when you start looking at other species. And so essentially translating these languages. Now, so we did a whole bunch of chemistry on these. These are various species are pests on everything from grains through to tree fruit and orchards, for example. And putting out some traps, we made these homemade traps because it turns out that these guys are escape artists, and so you have to make it very easy for them to get into a trap and very difficult to get out again because they will find their way out otherwise. But what we found is that we were getting very few of these insects actually inside the trap. So we looked at some possible problems with the pheromone. We rechecked the chemistry. We looked at dose and blend and so forth. We redid our trap design a number of times. And we were trying to think about other possible biological explanations for why this wasn't working very well. And what we came up with is buried in the literature from a group in Slovenia, which is just on the top corner of, as you go from Trieste in Italy, going east from there, a small country. And there was a guy there called Andrei Chokol who had determined that these insects were producing substrate-borne vibrational signals. That's a fancy way of saying that they vibrate on the plant. The vibrations travel through the plant and are detected by a receiving insect. And there's no sound component, there's no audible sound component to this. This is a true vibrational signal that just gets transmitted through the substrate. And these insects, they're actually producing quite a repertoire of these types of songs. And they're using them for mate location, for courtship, and males actually produce what are called rivalry songs, where they essentially sing to each other and basically saying, you know, buzz off or something like that to get the other guy to go away. Now, if this will work, I'm hoping it will. Can you just click on the one of the sound icons there? So this is what some of these songs sound like. These are male calling songs from this Acrosternum species. And if you could go to the bottom one now. So you can see that they're essentially speaking quite different languages. And you can see it visually from the sonogram that's displayed, but you get an even better idea just from listening to the signals. And okay. And that was the calling songs from males. So males are producing that signal and then hoping that a female will answer. And females also produce calling songs to see if there's a male around. If you can click on the icon again for me, so you can see what the... So that one's sort of fairly featureless, just sort of a buzz. And then the one below, if you would, please. And the frequency characteristics and so forth are, again, really quite different between them. Oops. Okay, and this is a male and female calling back and forth to each other. And this, I think, is my all-time favorite, if I got the right one in, if you would. I mean, come on, if you're a female bug, who could resist that? I mean, it's just... So, but I think you get the idea here that these insects are, they've got this communication system where it's really pretty much unmistakable if you've got the right sex and the right species and so on, because those songs are so different that it's difficult, it would be difficult for them to make a mistake. Now, so how do these signals work? It's a remarkable system, because what happens is they're calling to each other, and the male is able to find out where the female is by following the vibrational signal through the plant. And the way that he does that is whenever he comes to a junction, he straddles the junction here, and he's able to measure the time delay, or sorry, the direction of the signal by whether the signal is going this way and hitting the front legs first and then the back legs, or vice versa. So signal going down the plant, and that time delay is just a fraction of a millisecond, but the insect has the mechanical capabilities to be able to do that. It's truly a remarkable system. And so the way that we think the system works is they're using these chemical signals to get to the vicinity of, or to bring males and females together to the same vicinity, and then once they're on the same plant or the same substrate, then they start calling to each other using these vibrational signals. And as I mentioned, there's this sort of repertoire of different types of signals that they're producing. And with that, the capability of determining where or which branch the signal is coming from, if you think about it, it doesn't matter how complicated that plant architecture is, because as long as you can always choose the right path where the signal is coming from, you'll always be choosing the most efficient way to get to where the signaling individual is. It's a very efficient system, or a remarkable system that they've developed. And come back to Yogi, you've got to be very careful if you don't know where you're going, because you might not get there. And that was us in terms of thinking, okay, well, everything's got to be the chemistry. And then thank goodness for Andre, who got us, sort of made us see the light in terms of realizing that there were actually these two components to this system, the chemical signaling, and then at shorter distances, the acoustic signaling. Okay, another type of insect. These are, many of you may not have even noticed these. These are little tiny things. They look like a ball of white fluff on a plant, a couple of millimeters long. And this is the female. The male is entirely different. He's a little tiny guy. He only lives for maybe a few hours to maybe a day or so maximum. But many of these are very important agricultural pests, both from the damage, the direct damage they cause to plants, but they also vector a number of viruses and other plant pathogens. So the adult females are wingless and sessile. Adult males are non-feeding and winged. And the females then produce these sex pheromones so that the male will be able to find them. So about 2000 or so, we started working on this group of 4, which are the main pests, main mealybug pests in California on grapes. And in particular, the impetus was that this one is an invasive species that had recently gotten into California and was really wreaking havoc, in addition to having these other ones present there already. So just to give you an idea of how we go about collecting these pheromones, we grow or rear up the virgin female insects on squash here, and the squash are maintained in these large glass containers. We sweep clean air through, and there's a collector on the front here where we collect, it's a chemical trap essentially, that we collect the volatiles that they're producing and then extract those with solvent. So the first one we did was the vine mealybug, and this is a gas chromatogram trace, which basically each peak represents a different compound. The bottom upside down trace is just from the clean squash. The top one is from the virgin females, and you can see here that there's the peaks 1 and 2 are really clearly associated with the insects and not with the squash itself. So they're very likely candidates for the pheromone. And we fairly quickly were able to work out the structure of this compound, a slightly unusual monoterpene-based compound with an ester function to it. An interesting structure in its own right. The second one took us quite a lot longer, two more years, and the reason being that these insects produce very tiny amounts of these compounds. And the structure of this one was unusual enough and difficult enough that we could not determine the structure just from mass spectrometry and chemical tests alone. We actually had to get enough for a nuclear magnetic resonance spectrum on it, which at that time required a few micrograms. And Sharon Midland was the NMR technician who was very instrumental in helping me to sort out exactly which stereoisomer we had here of this new pheromone compound. The third one, long-tailed mealybug pheromone, again, took us another couple of years. And in this case, we used the whole battery of mass spectrometry, microchemical reactions to determine some of the structural features. And this was the first time we were able to use what is called microprobe NMR, which enabled us to push down the amount of material that we needed. And we were able to narrow the structure down to these two possibilities, where we could not be sure exactly where this double bond was. So we started out by just making the first one, and of course it turned out to be the wrong isomer, but that's fine because that meant that the other structure then had to be correct. So as chemists we thought, well, there are ways of being able to essentially just shift this double bond from here to here. So we tried that since we already had that structure, and so this was the sequence of synthetic steps that we were going to use to do that. And my postdoc at the time, Jardel Moreira, is a very good chemist from Brazil, went through this. And what we found was that instead of being able to shift the double bond over, we had done a 5-step synthesis to wind up with a less than 10% yield of our starting material. And what happened is that instead of getting the oxidation in the place that we thought we were getting it, there was actually a rearrangement that occurred during the oxidation, and so it put the double bond in the position that we actually had wanted it to be in, and then the next steps were actually to take that double bond out and put a double bond in here. So at any rate, less than 10% yield of starting material. We were all rather embarrassed about that, but these things happen. Now the last one turned out to be the most difficult one, and this was where we used coupled gas chromatography, the separation method with electroantennograms. So reminding you, these males are about 1 millimeter or so long, and this is the head of a male, and the antennae here are stretched across between these two electrodes. And to give you an idea of the scale, the gold wire here is 0.2 millimeters in diameter. So you can imagine how tricky it is to set up one of these preparations. And my guy Steve McElfresh is just superb at doing this. And to show you the power of doing this, this is what the crude extract of the pheromone looks like. So you can see there's a whole mess of peaks here. We would have no idea as to which one of these was actually the pheromone. And then the bottom trace here shows the response from the antenna. And you can see that it's absolutely unmistakable that the antenna is responding specifically to that peak right there, even though it's a very minor component in the overall extract. So it enabled us to find exactly which compound in that mess we needed to focus on to identify the pheromone. And finally identified it with the help of Bruno Figadère, who was a sabbatical scientist who came from France to work with me for a year. And took 6 years of extracts, fractionated by HPLC first. It was still too impure to use, fractionated again by PrEP gas chromatography, isolated about a microgram, and then finally solved the structure by microprobe NMR. And to actually use these compounds then, this is a sticky trap, the typical traps that growers use for being able to monitor a number of insects. And here is a rubber septum. It's about a centimeter long, impregnated with the pheromone, and then each of these little dots here is a male mealybug which has been caught. And these lures are incredibly active. So 5 micrograms of material will last for several months out in the field. It's just extraordinary how sensitive these insects are to these compounds. And again, to give you some sense of that, if you have a gram of the pheromone, that would be enough to make 200,000 lures. So even though the compound is expensive to make, it's well worth it in terms of having a commercializable product. And mating disruption of vine mealybug, at least, has been been used in California over several thousands of hectares of wine grapes in California, and in particular the high-value wine grapes, because the mating disruption technique can be a little bit more expensive than using insecticides, but it also can work extremely well. So little things are big. Back to Yogi again. Okay, again, another system that we've worked with, and this is a system we're still currently doing a lot of work on, is the cerambycid pheromones of cerambycid beetles. And these are a variety of them. This is an invasive species that got into California. This one is North American. This one is also North American. Well, these three are all North American here. Wonderful insects. Approximately 30,000 species described, so there's lots of them worldwide. They're some of the most damaging insect pests. So they can get into forestry and plantations, orchards, ornamental trees, lumber and wooden structures. They're wood borers, and so the larvae basically are boring in anything sort of woody. And because of that, they're highly invasive because they get into packing cases or the pallets and things like that that get shipped around the world. And so they emerge in the country that they've been shipped to and can establish populations, and then you've got a major problem on your hands. They're also a very diverse group of insects. This is, doesn't even fully reflect the range of sizes, but just from this alone you can see there's some really small ones to very large ones, and the largest ones are I think about 10 centimeters or so long, so not what you want dropping down the back of your neck in a tropical forest if you're out there, for example. Now, we had proposed, or Larry Hanks, my longtime collaborator in this, had proposed three possible scenarios. First of all, you can have strong attraction to volatile pheromones. And in 1999, when he wrote this review article, there were no known examples of that. Weaker attraction over short distances, there were several examples of that, and then no apparent use of volatile pheromones at all. So what we started out to do was to try and get some sense, at least, of how important these compounds might be overall within this large insect family, to get an idea of the types of chemistry that they were using, and to see if we can develop some predictive power so that if we got an invasion of a new species into North America, or anywhere in the world for that matter, just by knowing its taxonomic position, would we be able to predict what types of compounds it was using as pheromones, so that we could rapidly develop a method of detecting it and using it in eradication projects. So, some basic science then, looking at insect behavior and chemotaxonomy. Can we use these chemicals to help trace the lineages of these insects, for example? And very strong applied push here, particularly to be able to develop tools for detecting invasive species as soon as possible after they have entered a new country. So the initial plan was to synthesize and screen a library of compounds based on the few known structures that there were, to basically look at those and then think about, okay, well, what are the logical modifications we can make to those, change the chain length, change the functional group positions and things like that, and screen those just to see what happens. And a second approach where we wanted to target specific species where there was absolutely nothing known in that particular subfamily or tribe, for example, to fill in some of the taxonomic gaps to be able to give us a new toehold into an entirely new group of these insects. Now, what was known from the 6 or 8 examples that were known around 2000 was that there was this highly conserved structural motif where you've got a carbon chain here which is 6 to 10 carbons long in total, so R is 3, 5, or 7. And then you have an oxygen on carbon 2 and carbon 3, and those can be either alcohols or one ketone in an alcohol, or in some 2 ketones. So really strong biosynthetic parsimony seemed to be the order of the day for the small group of cerambycids for which pheromones were known around 2000. So that's what we based our library on. And so in 2004, then when Jeremy Allison and co-authors did this review article, there were pheromones or sex attractants identified for less than 10 species, and they were all from one subfamily. There are 8 subfamilies in total in the cerambycids. And the traps and lures were not optimized either. And this, in fact, had contributed to the problem or the idea that people thought that these insects were not using pheromones, or very few of them were using pheromones, but that turned out to be quite different. So now, 11 years later, we know pheromones known or suspected from several hundred species in 5 of the subfamilies, and we've been able to optimize the traps, optimize the lures, and optimize just generally how to use these most effectively to be able to track these insects and find out when they're coming in, where they are, and that type of thing. Now, I'd like to just tell you one story about one of the targeted identifications that we did. This species here is called Prionis californicus. It's actually extends all the way from Alaska down into Mexico, so it's very broadly distributed on the west coast. And to give you an idea, I think most of you know click beetles. This is a click beetle, and so this is our Prionis. So these beetles are about as big as your thumb. They're really massive, burly guys. And you can see here, this is a male, and you can tell because he's got this very elaborate antenna, which is what he's using as a sensory device to be able to detect the signal from the females. So why do we choose Prionis californicus? It's a major pest of hops that is grown up in the Pacific Northwest, so Idaho, Washington in particular. And why is that important? Well, from hops, you go to beer, and I think if there are chemists in the group, I think you would agree with me that chemists think that beer is one of the 4 major food groups. And to give you some sense of really how important this is economically, though, this map shows the beer consumption by state across the states, and the winners, I think, are Montana, where the Montanians drink an average of 44 gallons of beer per person in the state. That includes men, women, children, everybody. So you can imagine there's a lot of beer getting drunk. And also, as you might imagine, the state where the least beer is drunk is Utah, and I won't belabor that. But this is, it translates up into Canada as well, because this is beer consumption in Canada broken down by provinces. And for some reason or other, the people up in the Yukon seem to drink inordinate amounts of beer. Not sure why. But my point here is that this is a huge industry. So the hops that are grown in the Pacific Northwest is about $500 million in that area alone. And there are hops grown in other areas as well. So it's a major pest of this crop, and they really wanted to do something about it. Now, Jim Barber, who is the guy in Idaho that we worked with, the entomologist up there, he discovered that the females have this, this is the egg-laying apparatus, the ovipositor, and there's this odd little gland that pops up here when the female puts out her ovipositor. And Jim was able to determine this is probably where they're producing the sex pheromone that attracts the males. And it's a very powerful attractant. It attracts males from hundreds of meters away. Extraordinarily powerful. And in order to be able to get enough of these beetles to work with, because they live in the roots of the hops, and so what Jim and his guys did is every few years the hop growers have to actually pull the hops out and leave the land fallow for a year or two, because the infestations of these beetles get so bad that they can't grow a commercial crop. So his guys went along behind a grower who was pulling out the hop plants, and they harvested the larvae of these beetles and put 1 larva per bucket with a piece of hop root, and essentially established a beetle farm here where you can only put 1 per bucket because they're cannibalistic. They will attack and eat each other. And so one beetle per bucket and then overwintered them with a watering system to make sure that they didn't get too dry and so forth. Adults emerged in the spring with an emergence cage over the top of each one of these buckets to catch that emerging adult. And we were able to get something like 150 beetles to work with per year. And the pheromone turned out to be really very difficult to be able to get enough to work with. So the typical things that we would do, solvent extracts didn't work, collection of odors from live females also didn't work. And to give you a sense of this, this is the gas chromatography trace, and this is the electroantennogram trace from these. And you can see here that this large peak doesn't correspond to anything that we can see in the gas chromatograph trace. It's, in other words, our analytical instruments are far less sensitive than the insect's antenna is. It's detecting amounts that we can't even see in our analytical instruments. So finally, after really struggling with this, Steve McElfresh, my technician, worked out what's called a solid phase microextraction. It's a device for sampling on small scales, and was able to get enough that we could actually see the compounds. And you can see here the strong antennal responses we're getting. This is the main compound, and then there were some analogs here that also gave antennal responses as well. And we were finally able to identify this structure. It's, again, an unusual compound for an insect pheromone because it has this free carboxylic acid, has 2 chiral centers here. But very fortunately, there are 4 possible stereoisomers then, but the insect, if you make a mixture of all 4 of those, which is the cheapest way to do it, the insect is not inhibited by any of the unnatural stereoisomers, so you can use this mixture of 4 stereoisomers to attract it. And it turned out that as expected, that this structure is highly conserved as well, so that prionic acid, as we called it, it attracts these 2 species in Georgia, these 2 species in Arizona, these 2 out west. In Europe, it attracts also another Priona species, Coriarius. And recently, just this year in fact, we discovered in China, this Doristhenes species is the major pest of sugarcane, which has grown on more than 1 million hectares in southern China. So it could be extremely useful for being able to manage these insects in China, as well as all of these species here, some of which are feed on the roots of fruit trees and things, in addition to the Prionis californicus, which is on the hops. One of the other things we're actually looking at now with these insects too is that the insects are covered with a layer of what are called cuticular hydrocarbons, which are primarily there for water balance so that they don't desiccate or don't get waterlogged, as the case may be. There are several reports of these being used as contact sex pheromones. That is, once the male and female are together, the male touches the female with his antenna, and just from that, the chemical blend that's on her cuticle, he's able to determine that she is the right species, first of all, and that she's a female. But there was also some indication that males could actually use these as what are called trail pheromones. That is, they could essentially follow the footprints of a female wherever she had been as a secondary method of being able to find her. So what I'd like to do is, there's a video clip in here, if you could. Great. And what you see here is the trail that's been penciled in here. And David Hall's collaborators, this was actually some work that was done in India, had basically painted a trail along that pencil line. And you can see here this male, he's going back and forth, never very far from it. So he really is following the trail, trying to find the female that he thinks is there. So we started investigating this as yet another method by which these insects are using chemical communication. In this case, though, it's a contact thing, non-volatile compounds that they're using instead of the volatile ones. And while we're talking about hydrocarbons, I'd also like to just show you this. This was some work that I did with a fantastic field biologist called Leslie Saul-Gershens. And what this is this bee here. This is a male bee. a solitary bee species that lives out in the desert in California. And this sort of brown stuff that's on him are the first instar larvae of a parasitic beetle species. And what the beetle does is these first instar larvae, they parasitize bees' nests of a particular species of bee. And so they have the problem of how on earth are they going to find the nest of these bees out in the middle of the desert. And the way that they've done it is they've mimicked the pheromone that the female bee produces to attract the male. So what they do is these little guys, they climb up to the top of a plant and they sit there and they essentially make like a female bee. The male comes along. As soon as he gets close enough to them, they all instantaneously leap on board him. He flies off. He finds a female bee and mates with a female bee. They transfer over to the female bee, and she takes them down to the nest. They disembark down in the nest and they parasitize the nest and feed on the pollen stores and the bee larvae that are down there. So it's an extraordinary co-evolutionary study, and it's all based on hydrocarbons. This is the adult beetle. It's a meloid beetle. These are actually quite interesting insects in their own right because they're chemically defended by a compound called cantheridin, which is commonly known as Spanish fly. It's the ancient aphrodisiac or claimed aphrodisiac. It's actually very toxic, and these beetles use it to defend themselves. And if you could show. Now what this shows is a male bee coming in, and see how fast that was? That was literally 200 or 300 of those larvae transferring to that male bee in a split second. And then he sort of falls to the ground and rolls around, and they distribute themselves over his body because he's covered with these hairs. And then as soon as his wings are clear, he flies off and finds a female carrying this lethal burden with him to take down into the nest. Now, this shows the hydrocarbons that the female bee uses, and the triangulins, which is the name for those first instar larvae. This shows the mimicry, and so you have the male hydrocarbon profile here on the bottom, and you can see that the female one is a lot more complex, and the triangulins then are using a subset of those compounds produced by the female, but that subset is enough that they are able to attract the male bees in, and as soon as he's there, or as soon as he gets close, then they all jump on board and get carried off. So remarkable coevolution, how these beetles, these parasitic beetles, have solved the problem of how to get down to that nest. They essentially like calling a taxi almost to come and get them and take them there. And again, to finish with Yogi, I think we're lost. Yes, but we're making great time. And I'd like to just, I think, finish with that in terms of the young people in the crowd to not mistake activity for progress. Because it's very easy to generate data, but you need to make sure that the data actually means something in order to really make progress. And I'd like to thank some of the important people who've played a role in this. First of all, Cam Oschlager, who is my supervisor at Simon Fraser. And Cam is still very much active. For the last 15 years or so, he's been running a pheromone products company, one of the most successful in the world, down in Costa Rica because his wife is from Costa Rica. And I just saw him a couple of weeks ago at the Entomological Society meetings, in fact in Minneapolis. This is Steve McElfresh, my lab wizard guy, out chasing moths somewhere in the desert in California. He's the guy who's done a lot of the electroantennogram work. He's really just an absolute whiz at making these preparations. And then Larry Hanks was a postdoc with me, started as a postdoc with me back in the 1980s when I first went to California. He has been at University of Illinois since about 2000 or so, and we've been collaborating together for the last 11 years on this whole cerambycid story, which is continuing to unwind. And of course, to thank the people who have funded so much of the research over the years. It's a combination of funding from private sources, industry sources in California, and federal sources of funding in the US, and even, in fact, from the British Columbia government in terms of the spruce conework, for example. And finally, a special thank you to Mary Catherine and James Fallona here today for allowing me to come up and share a little bit of this fascinating science with you. I still love it. I'll love it till my dying day, even though I don't like the paperwork that goes along with it, but that's just part of the burden. So thank you very much.